Hello everyone, welcome to this wonderful session, that is part 5 this is of chapter 2 biological classification. In this part we will discuss fungi, their general characteristics and different classes of fungi, that is 𝐏𝐋𝐀𝐘𝐋𝐈𝐒, then 𝕍𝕀𝔻𝐻𝐿, 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 different examples of fungi and economic importance of fungi. This is part 5, chapter 2, biological classification, unit 1 of 11th standard, that is diversity in livings. In this we have to discuss kingdom fungi. You know, dear students, that already we have discussed the two kingdom of 5 kingdom classification. The one was Monera, and in the last class we have discussed that is kingdom Protista. Is it? Now we'll go for kingdom fungi. You are familiar with fungi. You have seen that is fruit rot. You have seen that is sometimes that the fungus used to appear on leather, sometimes on beds, sometimes on our that is food also being spoiled by fungus. So study of fungi is known as mycology. These are heterotrophs, not able to make their food because these are non-green. These are heterotrophic organisms, and very important point, they have great diversity of mycelial structure and habitat. The fungus may be unicellular like yeast, or they may be that is multicellular, that is large structure, as you have seen these are the mushrooms. So they have lot of diversities. Fungi prefer to grow in warm and humid places. Humidity is essential, and that is a good temperature is also. If you will keep something in refrigerator, you know, it will not be spoiled by fungus because here the temperature is low. The fungus prefer to grow in warm and that is humid places. Fungi are usually filamentous. They consist of long cylindrical structure like this that may be branched. It is thread-like and known as hyphae, which form a network that so many hyphae may be put together like this, and this net of hyphae is known as mycelium. That is known as mycelium. The most of the fungus, these are multicellular, but the yeast is unicellular fungus. You have to remember, you no doubt that yeast has been that is placed, classified now in kingdom Protista, in 5 kingdom classification, but even then the most of the character of yeast are similar to fungi. Therefore the yeast we have to study even now along with other fungi. Mycelium will be of two types. Cenocytic mycelium, you hear that this is the mycelium, so many nuclei are present. There is no septa, that is it is multinucleated. Aseptate mycelium, and this type of mycelium is found in case of that is 𝐏𝐋𝐀𝐘𝐋𝐈𝐒. 𝐏𝐋𝐻 having two groups, 𝐿-mycetes and zygomacetes. You have to remember that is mycelium of the 𝐏𝐋𝐻 is that is multinucleate and aseptate. Otherwise it may be septate also. That is, this is the mycelium, the septa is there and inside the nuclei are there. That is known as septate mycelium, that is found that is in 𝐿-mycetes, basidiomycetes and deuteromycetes. Is it right? Now the cell wall of all fungus, this is not made of cellulose, this is made up of chitin and polysaccharide, also known as fungal cellulose. Cellular structure, that is important point here, is unisister Golgi bodies. You know, Golgi bodies in case of other eukaryotic cell, they have so many cisternae like this. These are lying parallel to each other. Tubular structures are there, but in case of fungi, there is only one cisternae. This is known as that is unicisternal Golgi bodies. Nutrition is heterotrophic, non-green these are. They may be saprophyte, you know the meaning of this. They depends on dead decomposed matter. They decompose the organic matter. They may be parasite disease causing one, or they may be symbiotic one. The reserve food material is glycogen and that is oil. Okay. That is the asexual reproduction. It may be by fragmentation, by fission or by budding. Fragmentation easily. This is the filamentous algae we have here. This fungus, hyphae, that may be divided into so many segments. Each will develop into the new one. Fission, that is just like yeast this is. That is fission it will divide into two, just like that is fission will be formed. That is two structures will be formed. Budding means that the mother will be there and the protuberance will arise, the bud will arise. Occurs by formation of asexual spore. By mitosis division these are formed, therefore known as mitospores. That is, other asexual spores are, that is zoospore. These are produced endogenously and these are motile. Let this is the cell here. You see, let this is the cell. That is, this cell is known as zoosporangium. Inside the zoosporangium, that is mitosis division will be there, and as a result of which the spores will be formed, and these spores will be released, and these spores, they are having the flagella. Flagella are on lateral side. You have to remember, lateral flagella are there, and the flagella are heterocont. That is, two flagella are different. One is tinsel, one is smooth. That's the difference in zoospore, that is of fungi and algae. These are motile, these are heterocont, maybe isocont sometimes, and as in case of that is Saprolegenia syncytium. These are not formed in all. These zoospores are formed only in lower fungi, that is known as aquatic fungi. Otherwise, the flagella is absent. Is it right? Sporangiospores, that is, this is the sporangium. These are also formed inside, and this will be known as that it will burst and the sporangiospores will be released. These are also produced endogenously, and these structures are known as sporangiophore, that is formed in case of Rhizopus and in case of Mucor. Is it right? The third important spore is conidia. Conidia, see that this is the tip of the mycelium, and tip of the mycelium that cut off this, that is series of conidia. They will be formed exogenously, and like this you see the arrangement. The younger one and the smallest one will be close to this conidium, while the that is at far, that is away from conidium, that will be largest and the oldest one. Such an arrangement is known as, that is basipetal arrangement. What is this? Basipetal arrangement. Can you identify? Zoospore is motile, endogenous. Sporangiospore is non-motile, endogenous, while the conidia is non-motile, and that is a chain is produced. These conidia are non-motile, formed exogenously outside the conidium, as in case of that is penicillium. Is it right? Sexual reproduction, very important. Sexual reproduction is completed into three stages, into three steps. And these three steps very important, that is plasmogamy, followed by karyogamy, followed by meiosis. After plasmogamy, a distinct stage called dikaryophase is observed in Echomycetes and Basidiomycetes. You have to see what we mean by. These are the higher fungi. In higher fungi we have, this is higher, that is fungi, Echomycetes and Basidiomycetes. This is the mycelium, that is vegetative mycelium. They are having one nucleus, that is per cell, one nucleus. One will be of plus strain, the other will be of that is minus strain. That these two come together and they will get fused like this, and here in this cell the two nuclei will be there, plus and minus. This stage is known as, that is dikaryotic cell. In due course of time, from this dikaryotic cell, other nearby cell will also become dikaryotic one. That was the parental one, dikaryon, and that other nearby cells will also become dikaryotic one. This is known as monokaryotic hyphae, or this is also called as, that is primary mycelium. It is also called as primary mycelium. We are discussing important point, and this stage is called as dikaryotic phase. Karyotic phase, is it? And this is known as dikaryotic mycelium, also called as secondary mycelium. So secondary mycelium has two nuclei of opposite strain in a cell. We can denote it as N and N. We can denote this dikaryotic phase N plus N. This is also N plus N. During sexual reproduction, that the two will get fused, and here a diploid stage will be obtained. that will be called as 2n. Secondary mycelium, primary mycelium, monokaryotic one or dikaryotic one. You have to remember. That is, zygote is formed. You see, plasmogamy, a distinct stage, dikaryophase, I told you. It is observed only in Echomycetes and Basidiomycetes. In Phycomycetes, this is absent. Meiosis, zygote has been formed. Now in this zygote, that is meiosis division, will achieve again this haploid stage. Zygotic meiosis is present, very important point. Embryonic is absent and have haplontic mode of life. For most of the life cycle, the fungus is having, that is haploid stage, that is only zygotic phase is there, which is 2n. That's why haploid dominate, it is called as, that is haplontic life pattern. Is it right? Come to the next. Site of meiosis, in general we are discussing, it may be zygospore, that is, it is also 2n structure. Zygote or oospore, all these are, that is diploid structure. So meiosis can take place in zygospore depending on the type of fungus, type of group of fungus, zygote or oospore. That is, in case of oomycetes, that is oospore or zygote, while in case of zygomycetes, that meiosis will take place in, that is zygospore. Is it? In case of Echomycetes, it will be in eschus. Eschus is a bag-like structure here to see. Here this is that is zygote is there. There will be reduction division, or that is mitosis. And in eschus, the spores will be formed, which are linearly arranged usually, escospores. While in case of Basidium, that this is the Basidium, this is the zygote, 2n, this is zygote here, and that is reduction division. In zygote, that will make that 4 haploid nuclei, like 4, and little later all these will be shifted outer side. So these basidiospores, this structure is known as basidium. This is called basidium, and these spores are called as basidiospores. So can you remember basidio, that is spores these are. Okay. So basidiospores, these are exogenous in producing Basidiomycetes. Escospores are endogenous, produced in Echomycetes. You have to remember, right? So sexual spores in fungi, yes, diploids are, as I told you, diploid, this is zygospore. In case of Zygomycetes, oospores, and the haploid one, Escospores and Basidiospores. Is it right? One of the aspects of the fungus, that is, we are discussing general characters, is over. Come to the next now. Classification of fungi, that is important one. How the fungi are classified? When the question came, fungi are classified on the basis of pigments. On the basis of pigments, photopigment. Is it right? Is it right? No. Because the pigments are completely absent in fungi. That's why the pigment cannot be basis of classification. The fungi classification, it is based on mycelium, that is mode of spore formation, and fruiting bodies. Mycelium, right now you have seen that the mycelium is aseptate, or that is the septate one. Is it? Mode of spore formation, this is conidia, this is ascospore, or this is anything else. That is what type, and fruiting bodies present. So the true fungi, they have 4 classes. What are these classes? Come on. That is, one is Phycomycetes, that is known as, that is lower fungi. One is Echomycetes, also known as, that is eschus, or eschus are formed in this, also known as, that is sac fungi, bag-like structure is produced. Basidiomycetes, here also known as, that is club fungi. Both these together are known as higher fungi. Higher fungi, and this is Deuteromycetes, in which sexual reproduction is absent. Here is sexual reproduction, that one is absent. Therefore, this is not a perfect group. This is known as, that is imperfect group. This is not complete one. This is known as imperfecti, fungi imperfecti. Is it? You see here, now we have to say class, these are the level of classes we may call. That is, we have 4 classes now. Class Phycomycetes, yes, Phycomycetes, that is, this is the lower group of fungi. Then Ascomycetes, Ascus or sec fungi. These are the higher fungi. So we have to discuss one by one. That is Phycomycetes first, Ascomycetes, and so on. The first one is Phycomycetes. Come on. Phycomycetes, these are aquatic, found in aquatic habitat, and these are also found on decaying wood. These are also found in damp places, or they are also obligate parasite on plants. That is, lot of variation is there. Important point here is mycelium of the fungus. That is, I told you, in case of Phycomycetes, the hyphae is like this. It is synocytic one having so many nuclei, and it doesn't have, that is, septa. So this is known as, this is multinucleated. This one is multinucleated, and this one is, that is, aseptate. This one is aseptate mycelium. Synocytic. Synocytic means multicellular in case of Phycomycetes. Okay, I told you, in case of Ascomycetes and Basidiomycetes, the mycelium is septate. Now see, Phycomycetes, the lower group of fungi, it has two subclasses. One is Oomycetes, known as algal fungi. The other is known as Zygomycetes, that is conjugation fungi. Is it? Now see now, algal fungi, Oomycetes, this is close to algae. That's why these are found in aquatic habitat, or these are also, that is, on decaying wood. These are, right, saprophytic, or they may be either, that is, saprophyte, found on dead decaying matter, or a few are, that is, parasite also. We have to discuss. Hyphal wall contains cellulose. Do you remember? That is, all fungus, they have chitin. But this group of fungi, this is having, that is, chitin. All, that is, fungi, they have chitinous cell wall, but Oomycetes that have cellulose cell wall. Remember it. Asexual reproduction, this is also a unique feature. Both points are important. It is by zoospore, which are motile structure. Zoospore may have either one flagella or it may have two flagella, depending on the type of fungi. Sexual reproduction is by planogametic copulation or gametangel contact. Little bit detail is required here. Not given in NCERT, but you can understand it. Planogametic copulation means that the gametes are motile. This is the so-called plus and minus motile gamete. These are in the flagella. They will get fused and the zygote is formed. This is known as planogametic, that is, copulation. What is gametangel contact? Gametangel contact is that this is one, that is, structure known as, this is having the plus nuclei, and this is on other hyphae we have that this one is the having minus nuclei. The two will come in contact and the male and female gamete will be brought together. That is known as gametangel contact. Is it? Gametangel contact, that will be, as is found in case of albugo. The product of sexual reproduction and the site of meiosis is oospore. This zygote is surrounded by a little thicker wall, that is known as oospore. This is 2n. It will divide by meiosis. Reduction division will be there. As a result of which the spores will be formed. That is, you see, albugo candida, white rust of crucifer, mustard, that is well-known disease of crucifer, albugo candida. You can see that this is the leaf of the, that is, mustard plant. You are seeing that white patches are there, just like rusting is there, as the rusting is there on iron. These are brown in color, but here on leaf, that these are the white patches are there, that is known as white rust of crucifer by Oomycetes fungus, albugo. Will you remember it? The second one is Phytophthora infestans. You must have seen sometimes that the rotting in potato is there. Even in leaves the spots are there. That is known as late blight of potato. You must have seen rotten potato. It becomes black inside. It is because of the fungus Phytophthora infestans. This fungus has its record in history. The Great Irish Famine 1845 to 47 was caused by this fungus because that is, this is known as 𝐼⁻⁻ is there. That's why it is having a record in history. The next one is 𝐿𝐿 conjugation fungi. The other group of this subclass of the 𝐿𝐿. It is found in terrestrial habitat. Most of these are saprophytic. This is the fungus we have seen. You can do one thing. If you that have a piece of that is slice of bread, you that is put some water here and put some that is soil on this. You wrapped in a black paper, carbon paper, and put in a dark, warm and moist place. What you will observe that within a day or so, that is on this, the fungus will grow. Rhizopus or mucor will grow, and these fungus will be like this, that you can see at the tip they have that is headpin-like structure like this. Plenty, numerous, millions of the that is hyphae will be there in this fungus. So this is that is known as bread mold. You have to see terrestrial mold, saprophytic fungus. This is because bread is dead organic matter. It may be parasite also, but here in this group that fungus mostly are parasite. Hyphal wall, only this is the one which is having cellulosic one. Here the hyphal wall is made up of chitin or fungal cellulose, just like other fungi are there. Asexual reproduction is by sporangiosperm. I told you that time also. You see that these are the sporangia. Sporangia, how it will be formed? That these are the hyphae. Some of the hyphae, they will become aerial. Their tip will be swollen and it will become that round in shape. Here the nucleus will be transferred. They will divide by mitodivision and so many spores will be formed. That is known as sporangium, and that is these spores will be dispersed as you are seeing here. That is known as sporangiospores. So this is asexual reproduction. Millions of these spores will be formed. You can see a dusty powder is there. That is black powder. That is here even on this bread you can see in person. Sporangiospores. Sexual reproduction is by gametangial copulation. You have to remember what we mean by here. You see again, that is you have to see a little bit what we mean by gametangial copulation. Here children, you see that the two mycelium, one is of plus strain, one of minus strain, they will come close. They will make this protuberance like this. That is, one will be minus, one will be that is plus, and these are known as now gametangia. Each gametangia is having so many nuclei, and now this gametangia, they will fuse together. So here that is known as gametangial fusion or gametangial contact. In this structure, there will be so many plus and minus nuclei. In due course of time, they will fuse and they will make the zygote. That is known as gametangial copulation. Will you remember it? Detail is not there in NCERT, but you have to, that is gametangial copulation. Is it right? Then this structure which is formed here, this is thick-walled structure. This zygote structure, it is having that so many nuclei. That structure is known as zygospore, 𝐿𝐿. That's why the name is given, 𝐿𝐿. Product of the sexual reproduction is, that is zygospore, and this is the site of meiosis. The examples, typical examples we have, one is, that is Rhizopus, yes, stoloni. For this is, what we have, this is black bread mold, this is Rhizopus. Even mucor mucido, that is on dung. Dung is also that is dead organic matter, that is, this is known as spin mold. This is spin mold. And these two famous fungus of 𝐿𝐿, Rhizopus and mucor. Is it right? So children, you see, by now we have discussed that is important characteristics of fungi. Right now we have seen one of the class of 𝐿𝐿, that is lower fungi, two subclasses, 𝐿𝐿 and that is 𝐿𝐿, important characters. No need to narrate it now. Just now I told you. This is that is white rust. This is the blight of potato. This one is the mucor or Rhizopus we can say. That is on bread, that is slice, you can grow easily, and you can see that is sporangiospores, and that is how much number is there. Millions of the spores will be produced. Is it right? Then we have seen all this. That is like this, you see, characters of fungi, I told you, that is it? Then cell wall, about nutrition, why they prefer to grow in warm and that is humid places. Then zoospores, I told you all this structure you must be remembering. Zoospores are endogenous. These are also endogenous, but non-motile one is sporangiosspore. Conidia are exogenous and non-motile structure. Then site of meiosis, I told you. Classification of fungi: Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. In time to come here to discuss all of these. Then we have seen one of the class, that is Phycomycetes, and that some of the important fungi, that is its economic importance.